My name is Rešetar and this is Bastian Beyer. We are architects and research associates in two groups of the cluster Matters of Activity. Bastian is part of Weaving and I'm a member of the Material and Form Function project. We will present ongoing experiments with bacterial cellulose that are an outcome of a collaborative work which took place between disciplines of microbiology, material science and architectural design over the past year at the cluster. A catalyst for this collaboration was the Curtain project at the Humboldt Forum Labor, where the cluster has an opportunity to present the research in the new exhibition space in a public setting. And the aim of this project is to investigate the design methods that combine scale of microorganism with architectural scale through a performative installation that showcases the active materiality. And the Curtain project involves cluster groups Weaving, Material and Form Function, and Object Space Agency.
The Curtain is imagined as a changing experimental setting, a three-dimensional textile structure which combines plant-based cellulose crafted in rattan and bacterial cellulose pieces, which we will show some of them in our presentation. The curved multi-layered curtain is also partially a protection screen. And with the support of the Max Planck Institute for Colloids and Interfaces, we will be able to project the microscopic and time-based images that accompany experiments and display the intricate material structures and behaviors on otherwise imperceptible scales.
In our talk, we will elaborate on spatial concepts for cellulose biofilms. We understand these fibrous biofilms both as textile structures and as living organisms. The cultivated biofilms we are working with are complex entanglements of natural, technological, and material realms. In the beginning of our talk, we will highlight properties, production techniques, and the cultural and historic context of cellulosic materials. From there on, we will explore different aspects of the environment which are important for the activity of bacterial cellulose. Bastian will talk about nurturing environments and process of culturing and cultivating materials through microbiological activity. I will focus on the capacity of the bacterial cellulose to actively engage with climate and show how this can become a framework for novel environmental and structural design concepts. Finally, we will discuss design methods, experimental approaches, and examples from the recent work in progress, which will potentially open questions about directions, expanding these collaboration practices. In science, cellulose was first identified in 1938 by French chemist Beyer, who described resistant fibrous solid that remains behind treatment of various plant tissues. However, before discovering this sugar of the plant cell wall, cellulosic materials were deeply intertwined in thousand-year-old cultural practices, relying on materials such as wood, cotton, and other plant fibers as an energy resource, building material, and especially for textiles, clothing, and paper making. One of the earliest uses of soft cellulosic fibers might have been knotting, and from this invention, techniques were developed such as looping or weaving that enabled making items and artifacts such as baskets or nets to be manufactured entirely from fibrous materials.
As a skeletal component of plant cell walls, the polysaccharide cellulose is one of the most abundant organic polymers on Earth. Organized in a hierarchical structure, cellulose commonly appears in combination with lignin, hemicellulose, pectin, and other substances, forming complex structures with a distinct fiber morphology. Their structure and material properties are assembled through processes of growth in adaptation to the environment. Cellulose materials are harvested from various plant species with a variety of forms, from soft, fibrous to rigid. In contrast to monolithic materials such as plastics or metals, these grown materials are heterogeneously structured composites, but have been made into standardized products such as boards and panels.
The diagram shows the cumbersome processes of extraction and processing cellulose, which are energy and resource intensive and chemically laden, from source materials for the extraction through the use of chemicals to cellulose and its derivatives. These chains show complex industry-driven interdependencies, which place cellulose in the position of a raw material. So we need to get to it by processing other materials and then handle it as a resource for further derivatives. Each of the materials from this diagram, such as wood or straw, is a part of even more complex of fractionation in which material is further decomposed. Even though wood or cellulose are commonly considered being very sustainable, their production and extraction process usually comes with a certain environmental load and demands vast monocultures. So when we rethink materiality, we're questioning these dependencies and production processes. So the growth process involving material cellulose, however, at a small scale, has the potential of overcoming an understanding of cellulose as a raw material that needs to be extracted and then processed into something else. And so we can rather speculate on engaging the design in this process, not at the end of the production chain, but closer to the growth and the future material environments.
The growth process of bacterial cellulose in the lab is significantly simpler than the conventional processing, as there are no extractions from other materials, as the bacteria produces pure cellulose as a metabolic byproduct. This process needs little time and resources to grow. So this is the setup for static growth, which requires a nutritious medium and the bacterial strain called Comabacter xylinus, which utilizes various common sugars and, in contrast to photosynthetic plants, does not require light for its growth. The biofilm pellicle forms in the interface between air and the nutrient medium. The specific properties of bacterial cellulose are highly dependent on the culturing methods. Formation of bacterial cellulose generally occurs in several stages of materiality. The first one is when the biofilm is microbiologically active and the bacteria is permanently producing cellulose. And the second one is when the hydrogel is completely formed and the growth slows down or stops. In the third stage, the hydrogel is dried and only the cellulose containing the residue of the medium remains, and while the active growth has completely stopped.
Besides its biological complexity, bacterial cellulose is similarly interesting when it comes to material properties. It is in purer form than the plant-based cellulose, with higher water content and higher tensile strength due to the chain lengths. It possesses outstanding properties interesting for exploring structural and environmental design concepts in architecture, such as high water absorption, filtering capacities, high crystallinity, and the ability to withstand high tensile forces. It is a translucent, heterogeneous, skin-light, lightweight structure. Along with studies of bacterial cellulose in natural sciences and engineering, there is a recent research landscape with great interest in translating scientific knowledge and extending it towards speculative directions in architecture and design, with projects utilizing organic waste and energy infrastructure on urban scale, proposals for public growth, and the community knowledge or apparatus for growing cellulose into an endless role of material harvesting and pulling out the cellulose layer out of the nutrient liquid.
So all these examples work around the idea of cultivation. The term cultivation can be understood as a nurturing interspecies relationship, and it doesn't exclusively involve the human species. So some insects adapted the practice of cultivation much earlier than humans. One well-known example is what's called ant-fungus mutualism. In this example, leaf cutter ants harvest specific leaves, transport them over long distances into areas of their nest in order to cultivate a specific fungus to feed on it. Over millions of years, an inseparable relationship evolved. These constantly maintained and growing structures are at the same time food stock for the colony, as well as inhabitable metabolizing structures. The process of cultivation presupposes the management of resources, time, and environment. The forms of agriculture and farming, which developed over time, also advanced the sourcing of architectural materials. Fibrous materials, for example, could be sourced more easily and reliably from either cultivated plant materials or livestock providing wool or leather. Similarly, the development of forestation enabled easier access to wood with more homogeneous growth. So the refinement of food with the help of microorganisms, known as fermentation, can also be understood as a cultivation process. Through this process, we refine milk products, prepare sourdough, or rely on this process for brewing. Fermentation relies on the metabolism of distinct bacteria or fungi to transform food chemically and structurally, and for the preservation of food. This practice became an integral part of maintaining our own metabolism. This process uses raw material, for example, milk or grape juice, which are either naturally or artificially inoculated with a specific strain of microorganisms, kickstarting a microbiological transformation. As the microorganism feeds on the specific compounds in the medium, it generates the desired metabolic byproducts. This practice resembles a negotiation process which happens between human and microbiology. A set of boundary conditions, nutrients, geometrical or environmental conditions, is put in place by the human, which sets the stage for the microbiological actors to play their role. At a certain stage, the microhabitat, which is defined by these boundary conditions, is exhausted and the biological activity declines, or the process is being stopped. So cultivation processes are usually mutually beneficial relationships where one species tends a microhabitat for another organism. The caretaker benefits from a food source, and the cultivated species can develop in a protected and maintained environment. The field of architecture and design is mostly reliant on mineral-based, petrochemical-derived, or plant-based materials. However, microbiologically cultivated materials more recently have found their way into the architectural discourse. The materials have shown that they can substitute some energy and resource-consuming construction processes and allow for a circular materiality. This really marks a paradigm shift in the way architectural materials are produced. We shift from drawing inspiration from grown materials to designing with growth. The design process is less about designing an object itself, but more about designing the boundary conditions, such as nutrients, geometry, and temperature. These conditions determine the behavior of the organism, the structure which evolves, and similarly inform the material properties. In contrast to chemical or mechanical manipulation of materials, cultivated materials can be grown to match specific properties by influencing their immediate physiochemical environment. The environment quite literally makes the material.
So following on from Bastian's discussion on nutrition and cultivation and bacterial growth environments, I would like to talk about other aspects of interconnectedness between the material and the environment as related to climates. Architecture is often understood in terms of solid structures and tectonics, and the environment is something surrounding the solid matter, appearing immaterial. So what we would like to propose in the experimental work with bacterial cellulose is to understand the environment as equally material domain to structure, and how these dynamics, changes in temperature, humidity, not only transform the material but also become an integral part of structures. So we will tackle this duality, structure versus environment, in the example of bacterial cellulose as a highly hygroscopic material, sensitive and adaptable to changing environmental conditions, which in turn can also actively condition the surroundings. This duality was strongly opposed by an architectural theorist, Rainer Banham, who claimed that the environmental space, with its indeterminate boundaries, is no less architectural than the solid structure. In architecture, the cellulose, environmental technology, and materiality have intricately connected histories. So sensitivity of cellulose to humidity was an actual reason for the emergence of first systems for climate control in buildings. The apparatus for treating air was invented in 1906 when Willis Carrier was asked to find a technical solution to control environmental fluctuations of the interiors of textile mills and printing houses, where the humidity was causing deformations of paper and cellulosic yarns. Air conditioning enabled the full mechanical control of all aspects of the indoor climate: heat, airflow, humidity, with the promise of increased productivity and hygiene of the modern age. Not only it introduced standardized indoor climate, but it also detached architectural elements from engaging with the outside environment, resulting in generic insulating typologies. So, as in Bastian's talk on coexistence with microbial environments, we would like to approach contingency and the dynamics of the surrounding environment, involving heat, temperature, and humidity, and allowing materials to operate as open systems in flux and exchange with the outside conditions. Architectural examples with cellulosic and animal materialities dealing with these fluctuating environmental conditions can be found in various examples, such as temples in Tsukushima, whose construction is partly underwater, and it was built to be resilient to changing water levels, with parts that are exposed to weathering and gradually replaced over time. And similar logic of the structure made to last by being gradually replaced is the thatched roof. It's an indeterminate large number of straw fibers that create boundary that works more in a manner of material threshold than a barrier. Black tent is also an interesting example where a woven fabric of the goat's wool, porous in the dry weather and waterproof when it rains, thanks to the swelling behavior of the fibers. Material is inseparable from climate and the Yurok nomads' goat keeping and their knowledge of weaving. So all these examples are not based on the logic of control, but the ability of the material to work with surrounding environmental conditions and available energy, relying on material indeterminacy and its temporal behavior. Environmental exchange can be understood as a generative principle for transforming material structures, such as an example of these mud cracks formed in the process of drying and natural convection. Also, in the case of bacterial cellulose, the drying process starts to configure and reconfigure both the shape and the internal structure of the biofilm. And so the environment and the material become part of each other's composition in these processes, so that the environment is not an immaterial domain that surrounds the structure, but becomes incorporated into the material. Morphologies of drying are saving both elastic energy and the thermal energy that are passing through the material and are exchanged with the environment. So elastic energy is stored and released in the shape-changing process, and thermal in evaporation, a phase transition of water from liquid to gas, whereby the material is cooling the environment. And through the rehydration of the cellulose, it returns to its original shape, but actually the fiber alignment stays in the memory of the material. And in these examples, we can see that all the initial elements are simple and all the same, just connecting them in the wet state and exposing them to various drying conditions results in highly complex morphologies. The scale of the transformation is actually microscopic, so all these changes happen on the level of the fiber. The swelling process causes the biofilm to temporarily become gelatinous and then reverse to the dry state in which fibrous structure reappears.
The experiment started with hands-on, open-ended investigations of biofilms and their cultivation in the laboratory, and continued with different concepts that we could speculate on from there. As in the case of this biofilm, with the gradient of translucency, the question was raised, What kind of internal structure or growth environment led to such a quality? In a further step, analyzing the biofilms at a microscopic scale allowed us to understand their intrinsic behavior and how the structure can be articulated through the environment on a larger scale. The generation of advanced setups for scaling up the prototypes into larger structures was the next step in the design process. Here we developed 5 different strategies, which we will explain in more detail in the next part of our presentation. In the early exploratory stage, these different approaches were mutually informing each other.
The first attempt of the integration of the plant-based in microbial fibers was integrating perpendicular cellulosic thread during the biofilm growth. So we observed the bacterial cellulose growth in the presence of natural fibers and how the integration of multiple paper threads transformed the biofilm in the dry state from initially brittle structure to a flexible one. This setup was devoted to integration of various fibers into biofilms and creating a composite material which can combine different kinds of cellulosic material at the interface between the environment and the nutrients. This element is constructed in the way that fibers which are integrated can be interconnected into larger systems by knotting. So each node becomes a connection point for the neighboring films to build a continuous porous membrane. In the analysis of the internal structure, we discovered that fibrous plant-based cellulosic scaffold causes the biofilm to rearrange through drying. So these lines of stress that can be seen in the pictures are indicating local tensile strength of the material that is obtained in the drying process. Scaffold also behaves as a trigger for the growth. The pellicles start forming around the immersed threads. We developed a loom to wind the threads and enable the film to grow around them. And so after the growth, it results in skin-like texture with the details of the thread integration showing how the fibers are blending with and being coated with the biofilm. The drying process not only causes shrinking, but in some cases also strong adhesion. By overlapping individual sheets of cellulose and connecting them by magnets during the drying process, we were able to create larger formations without an additional connecting structure. Kurkuma was used for coloring with an intention to explore natural substances that could prevent biofilm from molding.
As Eva explained earlier on, this setup also emerged from the initial open experimental phase. What you can see here are the first experiments we did with the new setup. And as these first results were quite promising, we were quite motivated to continue to improve this setup. So we tried to improve the mechanical components to improve the extraction process. And through several iterations, a refined demonstrator evolved. In the installation, 2 worlds interfere with each other. A rather simple symmetrical mechanical setup interacts with a complex living organism. An override occurs between a very strict set of geometric boundary conditions and the complex behavior of a living biofilm. Involving living organisms in the design process means embracing biological growth and giving up control to a certain degree. The question arises whether contamination is a material failure or a microecology in the making. Experiments like this let us understand that we are surrounded by a multitude of microbiomes, which are inseparably connected with architectural spaces. So architecture is commonly perceived as a static condition, something permanent. With a lot of effort, materials have been engineered to be passive, withstand changes of environments with the least response. However, architecture is always placed in a constantly changing environment in the midst of processes of decay, decomposition, and restructuring. As some of the historical examples presented, architecture can be designed for and to react with changing environments through its materiality. A materiality in reciprocal relationship with its environment, which allows for aging and weathering, while at the same time reducing the need for artificial conditioning. In the same way, microbiology offers an opportunity to work with the dynamic conditions of biological matter. However, acknowledging this distinct authorship of microbes also means abandoning some engineering axioms which in the field of architecture dwelled over the last century, such as standardization and homogeneous and permanent materiality. Similarly, we can move away from mimicking growth structures to designing with biological growth. Hereby, the idea of engineered optimization is contrasted by the redundancy and non-deterministic behavior of biological systems. This demands design strategies to shift from static forms to designing relationships and dynamic processes. So thank you very much for your attention.
Thanks for your attention also from my side, and we're looking forward to the discussion.
See you.